Welcome back, students. Welcome to biology classes. Today we are going to see 11th science biology and we are going to see the new chapter, and that is chapter number 13, that is photosynthesis in higher plants, right? So today we are going to see the basic introduction of this chapter and then we will shift to the main topics of this chapter. So photosynthesis, right? So photosynthesis is a physiochemical process, right? Physicochemical process, that means the split the word into two: photo and synthesis, right? Photo means light, synthesis means to produce something, right? Now this photosynthesis is the mixture of the photo physio as well as the chemical processes, right? And this process uses light energy to synthesize organic compounds like sugar, right? So in photosynthesis, that is a process of photosynthesis, it uses the light. So it needs three things. First is the light, second is the water, and the third is the carbon dioxide. So in the presence of this three substrate, right, the photosynthesis is taking place, right? And it is going to synthesize an organic compound which is known as sugar, right? Now, what exactly is the importance of photosynthesis, right? So we will see that what is the importance. So first, the photosynthesis is said to be the primary source of food, right? That means whatever food a plant needs, that has been given by the photosynthesis. That is, upon the occurrence of the photosynthesis, the food is being produced inside the plant body. Next is it releases oxygen to atmosphere, right? So normally what happens is the plants in the daytime, when the light is present, at that time they intake the CO2 and they exhale the oxygen. That is, they release oxygen into the atmosphere. But in the night time, when the light is not present, for this reason, not like photosynthesis does not takes place. And for this reason, at night time, they release out CO2, right? So where if they in the daytime, if they release oxygen to atmosphere, then the amount of oxygen will be more in the atmosphere, right? Now see here, you can see this, that the water, right, this is absorbed from the roots, right? So what if from where the leaf is going to get the water? So that is, they are going to get the water from the roots, right? And the roots, from the soil, they are absorbing the water. Now, this water reaches to the leaf, as well as also the carbon dioxide enters inside this leaf with the help of the stomata which are present on the leaves, right? So this stomata, upon closing and opening, the carbon dioxide is entered inside the leaf, as well as the light is falling on this leaf. So this light, it acts upon the chlorophyll, which is a pigment, right? Green colored pigment. And this chlorophyll has a tendency to trap the light energy and make the food, right? So for carrying out the photosynthesis, it needs the first, that is light, then second water, and the third, that is carbon dioxide. The three of them, when it is present in the presence of the light, this chlorophyll traps the light energy and it produces the food, right? So when they produces the food, that is in the sugar, now in plants the food is stored in the form of starch, right? So this oxygen, whatever oxygen is being released over here, it is given into the air, that is, it is released inside the atmosphere, right? And the sugar, it is converted into the starch, that is the stored food in other parts of the plant. That means this sugar is converted into the starch, and this starch is the form in which the whole body part, or we can say in the other body parts of the plant, the food or the stored starch is present, right? And this starch, it turns like dark blue when you are exposing it to the iodine. So if you want to check whether starch is present or not, then if you add iodine to it, then it is going to show dark blue color, and this depicts, or this demonstrate that the starch is present, right? So this is about the photosynthesis. Now There are several early discoveries also which were taking place, right? So these are several names of the different scientists who performed several experiments to understand about the photosynthesis, right? So the first one was Joseph Priestley, candle with bell jar and mouse experiment, right? So he carried out uh, the candle with jar experiment as well as a mouse experiment, right? So he concluded that air is necessary for the growth of plant, right? Now here candle with a bell jar, right? Now see, a candle was been kept and a bell jar was kept on that candle, and a small at along with the candle a small potted plant was kept in that jar. So as the bell jar is now closed around the candle, right? For this reason the outer air is not able to enter inside, right? So from this like the plant was not able to grow because the it is not getting the proper amount of air from the atmosphere. So from that Joseph Priestley concluded that air is very necessary for the growth of a plant. Same way the mouse experiment was also being done in the same way, right? Now he discovered the fact that the plants restore oxygen in the air, right? That means the plants in order when they uptake the CO2 in order or in other way, it releases out oxygen in the air, and which is very useful for us, right? So this was about the first, that was Joseph Priestley. The second is Jan Ingenhouse, right? An experiment with aquatic plant in light and dark. So he performed an experiment with the aquatic plant, that means those plants which are growing inside the water, and those aquatic plant in light as well as in dark conditions, right? So he concluded that the sunlight is essential for plant processes that purify the air. Now he performed this experiment, right? That he allowed the aquatic plants to grow in the light medium also, where the light is present, as well as in the second medium where no light is present, that is the it is dark, right? So when the light is present at that time, if proper amount of sunlight is being provided to the plant, then the proper processes, that is plant processes, that is photosynthesis takes place, and the growth of the plant is proper. But when the light is not present and when the plant is kept in the dark, at that time what happened? The plant was not able to grow in proper way, right? Because there was absence of sunlight. So when the sunlight is absent, for this reason the process of photosynthesis does not take place. And if the photosynthesis does not take place, then the plant is not going to get proper amount of energy. And if energy is not given or not been produced, so then what happens? Then the plant cannot grow properly. So Jan Engenhaus concluded that sunlight is very essential for carrying out the plant processes. The third was Julius von Sachs, right? And he carried out experiment on the green parts of the plant which make glucose and store as starch. So he was the one who understood about the storage mechanism of the plants, and that is the how they store and in which form they store, right? So Julius von Sachs, right, he was the one who studied on the green parts of the plant, that why the green color, right? Why the leaves of the plant is green in color? So it was due to the presence of the chlorophyll, and this chlorophyll was responsible for producing the food, or that is the glucose, that is sugar, in the presence of three things. The first was the carbon dioxide, second was water, and the third one was the sunlight. So when the green parts of the plant, when the sunlight, oxygen and water, this sunlight, carbon dioxide and water, this three reaches to the green parts, at that time what happens? The light energy is converted into the chemical energy, that is the sunlight that is being absorbed by the chlorophyll. It is going to convert the light energy inside into the chemical energy, that is it is going to produce the glucose, And furthermore, this glucose is going to be converted into the starch, and this is the medium that is going to be stored inside the plant body, right? So Julius von Beck, he was the one to understand about the storage mechanism of the plants, and that is in the form of starch. The next is T.W. Engelmann, and he worked or he experimented on split light using prism into 7 colors, that is Vibgyor, right? So here green algae, that is Cladophora, placed in a suspension of aerobic bacteria. Bacteria were used to detect the sites of oxygen ovulations, right? So here green algae was taken, right? See here, split light using prism into 7 colors. That is one light, when you are exposing the one, like one monographic light, then it is exposed or it is forming 7 colors. When you are passing one single light into the prism, then it reforms the 7 colors, and that is, it injects out 7 colors, that is Vibgyor, right? This you have studied in your initial standards, that is standard 7 to 7 8 maybe, right? So this Vibgyor is what the 7 colors of the rainbow, right? Now what he studied is that he took a green algae, that the name of that green algae is Cladophora, and this Cladophora was placed in a suspension, right? So that was a medium in which aerobic bacteria was present. That is those bacteria which needs what? The oxygen, right? So in a medium there is presence of aerobic bacteria, as well as he also placed green algae Cladophora inside it. So this bacteria were used to detect whether the sites of O2 ovulation, that is, now see, this green algae, they upon carrying out or upon occurrence of the photosynthesis, they are going to release out oxygen, right? So as this bacteria is there, which are aerobic in nature, that is, they need what? Oxygen, right? So when the Cladophora is placed inside the suspension of the aerobic bacteria, at that time whatever oxygen is being released out by the Cladophora, right, that is all were detected by the bacteria which were present inside the medium. So this way we can understand that if, when a particular plant release out oxygen, only and only when the photosynthesis is taking place, right? So this way he, T.W. Engelmann, he worked on this, and he detected the sites of the oxygen evolutions, that is, at which sites the oxygen is being released out, right? And this way he understood that the photosynthesis is taking place. The last one, who was the Cornelius Van Niel, and he did the experiment with the purple and green bacteria, right? So there were, he took two different types of bacteria. The first one was the purple bacteria, and second one was green bacteria. And what he did is demonstrated photosynthesis is a light dependent process with hydrogen from H2O reduces CO2 to carbohydrates. He concluded that oxygen comes from H2O molecule and not from CO2. Finally, the correct equation for photosynthesis was discovered, and that is 6CO2 plus 12H2O gives C6H12O6 plus C6H2O plus 6O2, right? So now you see here that Cornelius Van Neel, he was the one who worked with two bacteria, that is purple bacteria as well as the green bacteria. He took this two of the bacteria, but now he demonstrated that the photosynthesis process, it is a light dependent process. Light dependent, that means only it occurs in the presence of the light, right? For this reason, this Cornelius Van Neel, what he did is he explained, right, that oxygen is coming from the water molecule and not from the carbon dioxide, right? So generally we have thinking that there are two molecules, right? So one is the hydrogen is coming from H2O, right? And it reduces the carbon dioxide to carbohydrates. So upon the photosynthesis, the light energy is converted into the chemical energy, and for this reason he concluded that oxygen is coming from the water molecule and not from the carbon dioxide. And for after this explanation, he gave a reaction, right? And this equation is 6 CO2, that is 6 molecule of carbon dioxide plus 12 molecules of water gives C6H12O6, that is the sugar, that is simple sugar, that is glucose, plus release of 6 water molecules, plus it releases 6 O2, that is 6 oxygen molecules, right? So 6 into 2, that comes to oxygen, how many? 12. 6 carbon, 12 oxygen, here also 12 hydrogen, and along 12 into 2, that is 24. So 24 hydrogen plus 12 oxygen. So it is when combining with each other at that moment, how many oxygen is there here? Here 6 into 12, 12 plus 12, that is 24 oxygen, as well as 24 hydrogen, as well as 6 carbon, right? So here you can see 6 carbon is balanced. H12 has came here, 6 into, it is 6 H2 in this, 6 into 2, that is 12. That is, here you are getting again, that is 12 plus 12, again that is 24, right? And here oxygen is also balanced. 24 oxygen should be balanced. 6 into 2, that is 12, plus here 6 is there, 12 plus 6, that comes to 18, right? So here also you can see that 12 oxygen is there, and here also 12, that comes to 24. So here 6 oxygen is already there, right? This 6 oxygen, this 6 oxygen, that comes to 12, and here the 6 into 2, that comes to 12, that is totally 24 oxygen molecules, right? So this way the whole equation was balanced, and this was been discovered by Cornelius van Neel. So today we are keeping this topic up to here, and from next topic we will see from the next lecture. I hope you are very clear with today's topic, and I hope to see you again. Thank you.